You probably heard terms like community biology, biohacking, citizen science, DIY biology. The truth is that in the past period, the low-level biology, the molecular biology, made some tremendous achievement and progress there, and things that were impossible before now became, possible. Think about, curing genetic diseases like cancer, for instance, saving the coral, making new materials that are lighter, cheaper, easier to produce, and non-polluting, solving the pollution, bringing back dinosaurs, and even extending life. So all this stuff is now possible. And guess what? Not only that's possible, but it's possible to be achieved by people like you. So when you do a project, of course, you can do a project by yourself, or you can do it in a group. Small projects, you can do them by yourself. So for instance, the small incubator that I designed, I did it completely by myself. For some of other projects, like the bioreactor, for instance, I had to create a group of people of, I don't know, 10 to 20 people that helped with that. So that allow everybody to bring different skills and different ideas. So that was great. Also, even in industrial research or in academic research, some projects require not too much resources at the beginning. And depending on the goal, they might require at certain phases some specialized resources or lots of financial resources. And they can go in there just by proving that different phases produced expected results and results that warrant, further investment. And it's the same thing in here. Knowing this, I was interested in this field, and a couple of years ago I started the community bio club. One of the things I discovered is was very easy to acquire, petri dishes, flasks, and other lab equipment, including reagents, which means the substances and even the bacteria that I wanted to, from a company called the Audin, for instance, and many other companies. And they were very cheap, like $5, $10, not too much. One of the things that were though expensive were devices. Some of the devices were into hundreds and thousands of dollars. And when you want to start with a project, you usually start with growing bacteria. And that happens with multi-million-dollar projects, like in skyscraper tower, ivory towers. They're very common thing because bacteria is easy to grow, it's fast to grow, and you can do all kind of genetic engineering on it very fast and very simple. So one of the things I discovered, as I said, it was that some of these devices were expensive and I didn't want to spend thousands of dollars. And I looked in the open source, which basically is open hardware, open source hardware, and I found several models there. And unfortunately, most of them were either not complete or they were not working. So together with a group of friends of mine, we decided to kind of solve this problem. So we ended up building a number of devices that are very affordable and they work because we are using them. Some of them we are using them daily. So the first and the most important one is probably the incubator. We end up actually doing 3 incubators. Another one that you can get the video how to do it, it's an inoculating loop, which is like a wire that you use to put bacteria in a petri dish. And the website is called pipettejockey.com, and it's a friend of mine, and he had a number of devices and other things that you need, but the inoculating loop is probably the simplest one of that. And it tells you where to get the materials and how to make it. Now, going back to the incubator. So we ended up making like about 3 incubators. One, it's a complicated one for liquid cultures. and it has its own controller and it's programmable and so on. We did another one that was programmable. But at one point, because some of them, the members of the lab, wanted to do their experiments at home because they couldn't come to the lab every day. And with the bacteria and with everything that goes with like organic life, usually you don't control their rhythm. They go by themselves. So we had a meeting on Sunday, and on Monday you had to go and kind of look at the culture, but some of them couldn't do that. So we decided to make this small incubator, and they decided to make them as cheap as possible and as affordable as possible, while not compromising on either the quality and the reliability and the capacity. So we ended up making these incubators, which are about, the capacity is about 7 petri dishes. If you really want to put them more, you probably can fit like a couple of more, and which is plenty enough for any experiments. And the incubators, they, depending on what variant you choose, it's about $25, which is very, very affordable. It's most likely the cheapest reliable incubator that you can ever use. The reason why we did that and we achieved that is because we used this thing called a controller that was already pre-built, and it had the display, it had the sensor, and it has the keyboard, and it had the drivers for the heating element, and it was only $4. So it's impossible to get cheaper with that. And that one, which we use, and not only me, but some other people too, is perfectly described. And the files that are in there, like I'm talking about how to cut the box, it's perfectly described in one of the videos called the cheapest, simplest incubator. Okay. So once you have that and you have the inoculating loop, then you can start growing bacteria. The other part, when you want to upgrade a little bit or you want to grow more bacteria, you would have to start with a liquid culture. The reason is, on a petri dish, you can grow bacteria only on the surface of the dish. In the liquid culture, it's like three-dimensional, so you can grow much more bacteria. The problem is that most of the normal research bacteria need some air and to not grow inside the liquid unless it gets air from time to time. So the simple solution for that, the normal solution, is to put them in a flask that will rotate. So that the media, the culture, moves around and comes in contact with the air from time to time, which is sufficient for the bacteria. For that, you will need an orbital shaker. And if you want to have a full solution, you want an orbital shaker that's in an incubator, so you can also control the temperature. Now there are maybe 1 or 2 designs of orbital shakers that almost work, but the problem is that the real orbital shaker has to work about 300 RPM rotations per minute, and that creates tremendous forces, and it usually doesn't work if it's 3D printed. Or as a matter of fact, I tried to 3D print it and it didn't work for more than a day, or something was cracking and so on. So we ended up making one that is reliable, and but it's using several components in such a way that works. And we tried it for about a week, and it was working very well. So that's another one. Once you go from the growing cells, which can be, mammalian cells, for instance, in plates, to in a liquid culture to obtain much higher density, which is necessary, for instance, if you want to harvest some protein made by them, because you cannot harvest that very well from a petri dish, or you want them for other purposes, the regenerative tissues or so on, and you need a much higher quantity that can to be usable. So that was another good project. Now, once you start growing in a liquid environment, you have to know how much bacteria you have, how many cells do you have there. And the simplest way, of course, you look at that, and if the solution is opaque, it means you have more cells. Now, one of the way to do that properly is to measure that with an instrument which is called optical density meter. And that usually is done with, you basically have a little glass container or plastic container, called a cuvette, where you put the media that you want to measure to see how opaque it is, and you cast some light using an LED or maybe a laser, and you have a sensor on the other side, and you read that value and you see how much it is. So for that, we created another device that is going to be, or it is already in a video, called optical density meter, which is very simple. That is 3D printed because there's no forces to deal with, no motors on it. And yeah, that would be probably about 5 plus 5, about 10 or 20 or $15, depending if you have the power source, which can be sourced from an older computer or can be used from an older computer, or if you want the display on the device itself or on the computer, which saves you another $5. Again, that is something that's tested, it works well, and we use it. And we use it even on larger or more advanced devices. These devices have been built for learning or for research. They can be used for simple cell culture or for advanced cell cultures. They can be used by professionals or by amateurs, and they have all been tested. We built more than 10 of each of these devices. They are affordable also. They are under $50. Now, we have more devices that we built and we are going to build. And we are going to add these in time whenever we can, and whenever we feel like they are stable enough and tested enough to be usable. So if you like to stay informed, you can certainly subscribe to this channel. We will put on this channel molecular biology information, and we will try to put new information, open source information, and tested information.